Welcome everyone to the third day of our international symposium on revolutionizing medical imaging and with computer vision and artificial intelligence from 22nd August to 25th August 2023, organized by MIT University. Our first session for today would be conducted by Dr. Rintu Kutum, Faculty Fellow, Department of Computer Science, Data Science, Emphasis Lab, ML and Computer Thinking, and Security, Trivedi School of Biosciences, Ashoka University. Dr. Rintu pursued his PhD from CSIR IGIB and joined Center of Excellence in Healthcare, IIT Delhi, as a project coordinator and a postdoc fellow. We are delighted to have you here with us today, sir. I now request Dr. Rintu Kutum to start the session. Thank you.
Hi everyone. I'll just try to share my screen. Hi everyone. So myself Rintu. So I'm a group lead at Ashoka University, and I primarily work in the area of computational health science. And thank you so much for giving me this opportunity to speak in the symposium. So today we'll be talking about the recent work that I have been doing in the area of computational cancer pathology. So before I move ahead, I would just like to mention that couple of things that which we work at Ashoka University is primarily driven by a lot of mentors, starting with Professor Subhashish Banerjee. So he is a computer science HOD at Ashoka University, and LS Shashidhara, he was one of the R&D department that we have at Ashoka University, where the most of the work that I have been doing in the cancer pathology is primarily driven by both of them. We do a lot of health science related activity and digital health activities along with Dr. Anurag and Dr. Ramesh Jain. So the the lab primarily focuses from the perspective of more of like a cellular ecosystem to digital health, and typically we believe in the framework of collective science through computational systems biomedicine framework. And we have 4 verticals which typically talk about to train scholars like UG, PG or PhD scholars. And also we do a lot of activities in the area of public health. And primarily the lab actually focuses on the the long-term version is on the looking into the computational spatial tissue biology perspective, where we do a lot of training in that, also do benchmarking of various machine learning algorithms. And the last vertical, what we focus in on primarily to facilitate patient-centric eco-informatics to assist clinicians and researchers and key partners, primarily in the area of health informatics. So before I move into today's topic, just I would like to give you my very short journey that I've been through in the field of research, especially in biomedical research, which is started in 2012, where at CSIR IGIB. Then since then I have never looked back to go back to my experimental expertise, but rather learned a lot of the stuff in the area of computation. And since then I've been contributing to computational biology, then machine learning, nowadays in the health science area. So during my PhD days, I really got a lot of opportunities to train a lot of scholars, which really actually helped me out in building next generation scholars in India, where we can do a lot of global challenges like health or bioinformatics, or in fact biomedical projects. Apart from this, I've been actively working with the open source community, where I've been part of different various organizations where we do training in R, Python, or Linux and other activities. Then apart from this, also we have been involved in a lot of the single cell genomics activities, where it had been highlighted in the Nature technology features also. So post my PhD, I joined IIT Delhi, where I primarily worked in the area of AI and healthcare, where whatever learnings that I did during my PhD on open source scientific computing, I really implemented those during my postdoctoral time. So this is one of the articles that which we published during my PhD postdoctoral time, which is to use machine learning and language models to, how we can use the spike gene, genetic variations, and can we actually represent the DNA sequence into more of like a nuclear, uh, what do you call as feature space, which will allow us to actually build models to predict or do surveillance of the SARS-CoV-2. So this is a simple framework that which I developed during the postdoctoral training, where we got a lot of the data from GSET, then automatically converting them into the frameworks and the workflows, where we actually take the sequence, then we remove the sequences which have a low confidence and also the low coverage, and also based on the collection dates, we organize them and QC and annotation of the data with the COVSEC algorithm. Then we also collected a lot of spike genes, around like 2.8 million data points. And then we use the language model to actually build the models. And typically in machine learning, or in fact deep learning framework, what I'm showing here is that, you should have a large amount of training data set, and also you can use that training data set to build the models. But nonetheless, also you have to do a real time, validations, which is in the green line, which we show that, that models is more robust, can be utilized for public goods. Then once we get the numerical representation of the spikes in, then you can use various methodologies to not only do the data analysis or the building models, but also you can use to visualize large scale data points using UMAP or in fact DSNE data visualization methodologies. So using the latent dimensions, which we call as the numerical space, we converted those into more of like a entropy based measures, which actually typically gives us the how variable that particular feature is. And with all monthly information it was converted, then we built models to actually predict the case load before the onset of the in fact the event itself. So we could able to predict like priorly in 2 months ahead that what will be the case load that might be in future, by just taking at the variation data sets from the SARS-CoV-2 spike gene. Then recently we at Ashoka, when I joined, so we started with a lot of the projects. One of the projects were in the area of computational cancer pathology. Today, whatever we'll be talking about is primarily in the area of digital pathology and also the deep learning application, where we have done into broad categories, which is on the one is on the triple negative breast cancer, and one is on the oral cancer. So I'll be talking about the latest work that we have done in the oral cancer, and we will have a more hands-on session demonstration on that particular data points and the learnings that we have done. Apart from that, we also work in computational genomics, where we have done like single cell biology or in fact system biology approaches for predicting ataxia or the host pathogen interaction with on TB side, and also evolutionary immunology. And primarily a lot of the works that which you see here, it's with a lot of collaborative effort. I typically participate in the computational part of it. The rest is like experimental part is taken care of the collaborators. So this is one of the work that after I joined Ashoka, I did work with public health expertise. Is most of them are from ICR Bhopal, ICMR Bhopal. So where we could actually build a lot of spatial data points, and can we actually predict the dengue epidemiology with the environmental variables. Then also I work with a lot of other people who are in the endocrinology department, especially for the PCOS and also the polymenorrhea, where I built machine learning algorithms to actually see is there any new phenotypes that we can actually gather. So now coming to my team members. So these are the small team members I have. Mostly are primarily not PhD students, but rather undergraduates or the people who are in ASP year. So the pathological team involves Akash, who is now graduated from Ashoka, but now he is in the 4th year doing his ASP. Then we have Sagarika. She is primarily come from biotechnology background, but she has a lot of interest in computational pathology, and she is one of the PhD student that will be joining the team. Then Somjit Basu is also a computer science and maths major student from Ashoka University. Amit is working in the computer science department for the computer science major. Abhishek is also a computer science major with more inclination to maths. Santosh and Karan both are in major in the computer science. So the team actually composed of a lot of expertise in, like, for example, Amit and Abhishek comes from very much of a tech expertise where they bring into the back end and the front end side. Shantosh and Karan are primarily involved in the machine learning part of it. Then Somajit is heavily interested in segmentation and temporal modeling of deep learning algorithms. Akash has been spent almost like 1 year in the lab where he has actually demonstrated a lot of and contributed to oral cancer and triple negative breast cancer. So now just, after the introduction, let's go to the topic of interest that we are doing today. So I will just give you a very brief introduction about what is digital pathology. So digital pathology is nothing but the technology that digitizes or analyze pathology slides for cancer diagnosis or prognosis, or it can be other disease condition also. Then when you see the workflows, the differences when we do a traditional way of like since till 2015, typically it used to have tissue preparations, then the glass slides were used to be couriered, and there used to be labs where the slides were analyzed and the diagnosis of the patient has been done. In the digital pathology workflow, typically what we have after we do the tissue preparation, then we actually digitize all of this tissue prepared one, and along with that we also uses a lot of machine learning and deep learning algorithms to analyze the digital slides, and automatically we can nowadays do a patient level diagnosis also on the report generation. So what are the benefits that typically the machine learning and gets in the medical diagnosis is that you can start finding risk factors, and also it also allow us to do an increase in the diagnosis more effectively, and also it reduces the unnecessary hospital visits, and so that the transactions become more swift. So this is slide which might be very interesting for you guys, so that it just clearly shows how we do we apply AI methodologies for digital pathology. So it started like the age of digital pathology really in application of especially the deep learning algorithm started in 2016, and it is primarily because the digital pathology data set were made publicly available, for which a lot of deep learning expert actually came into the field and started building a lot of sophisticated models which allowed us right now to actually have a more automated way of quantifying the pathological images. So in 2017, by the Besnor de et al., what they have actually done is like, can we actually predict by from the slide itself the metastasis breast cancer or not, whether they are in the benign or a metastasis. And most importantly, the part was the development of the data set. They started with 270 images, but they converted the images into more of like a mini sample set, which we will be demonstrating today in the hands-on session, where if you have a whole slide image, so you don't start building models on the whole slide, but rather take the segmented part of it, because the whole slide images become very, very huge in size, which makes the computer to handle the data is not so easy. So that demonstration also will have by Amit, who have built the or in fact build a structure to how or adopt algorithms which can readily be used to actually visualize in real time with very low resource constrained computing infrastructure. Then for the validation data set, they had around like 129 images just to validate how the model actually performance was done. So they have built a deep learning framework to actually predict whether there is the particular slide has a metastatics or not. Then for the validation, what they actually got around like area under the curve was around 0.99, then sensitivity was around 0.77. And they also did a retrospectively against by other group in 2018. They also did one more retrospective validation, and which give us the confidence that you can able to now diagnose or classify a particular sign more effectively. Then most importantly, apart from the developing the machine learning model, one of the most important component is the UXI development and validation, because the clinicians or the pathologists will not actually value this particular ML validation part, but rather they need to see or quantitatively see the slides and make a decisions. So it is not only the AI, but it is also with the pathologists which can actually interactively collaborate. And the technology is very, very important, that you should have a very robust UI for the pathologists, which will readily be useful for them to evaluate. Then also they have performed prospective validation for the AI and the digital pathology, and also the deployment and the monitoring of the whole stuff. And if you see the typical workflows that you see in the digital pathology workflow, typically with the same thing, you actually goes through a different approvals, like for example regulatory approvals or the integrating with the clinical workflow, and those become also very much challenging if you don't design at the very beginning the framework itself. You should have a, like, starting with the problem statement, but you should start from a more of the image acquisition to the a place where you build models. Then also you can actually do evaluation at real time by the pathologists. Then only we can actually take our AI based digital pathology technologies to a real world. So what are the applications of the digital pathologies? Typically, we talk about like digital image acquisition and the storage become very much swift, and also digital slide analysis and interpretation can also be done, very easily. Then automation and artificial intelligence integration of the cancer diagnosis or other part becomes very effective. And also it bring out a lot of accuracy that might be, exist inaccuracy in the when will be doing the manually by the pathologists. So the AI becomes more, a partner to the pathologist, rather than, the difficulties that might be or the inaccuracies that might result from a experience of the pathologist also. And most importantly, after we have those components for at least the clinical pathologies, but there are additional components also which make us life very much effective for at least cancer prognosis. So you can have a predictive modeling and also the risk assessment using same the AI in the digital pathology. Then also quantitative analysis of the tumor characteristics. I think this is the we are in the field where we are more interested in understanding that how every tissues of a particular patient actually vary across, and why there is any changes that at the ecological point of view of the tumor actually happens at the tissue level. Then that actually bring us to our more of a latest development in the personalized treatment plan that can be utilized by the findings from the digital pathology. And also it allows us to do a more of a long-term monitoring and also the follow-up of patients very effectively. But nonetheless, there are limitations are that which exist with many of the technologies. One primary one is the limitation of the lack of confidence in the diagnostics results right now, and also the opacity of the diagnostic process, and also the lack of interpretability of the diagnostics results. And the most important part is that, how we can make more interpretable AI systems for digital pathology is one of the very upcoming, or in fact that the field has been more concerned with interpretability of the results that a particular AI system develops. And second thing is the insufficiency of the validation of the AI, inconsistency in the practical use, and also the slow execution time of AI and lack of clinical workflow integration. So I think these things are primarily have been, now currently have been understood, but when we start with the building the models, or in fact the designing the whole workflow, we should actually take into this limitation in the account. And also simplicity of the functions are not so much widely been placed, then also the limited in the certain data and also specific tasks. So I'll just talk right now with our current work, that ongoing collaborative work between Ashoka University and the many hospitals, which was primarily led by Jamia Islamia, which is we call as the AI DORC, which is nothing acronym for an artificial intelligence based detection of oral squamous cell carcinoma. So this project has been patented, the whole workflow, the diagnosis by the doctor and the diagnosis by the algorithm that should be integrated, coupled, so that we can not make any decisions based on only algorithms, but primarily based on the expert point of view of the doctors and also the clinical pathologists, so that they can harness the algorithms or the AI methodologies easily to in their clinical workflow. So this primarily led by Tanveer Ahmed's lab at Jamia Islamia in New Delhi. So for this, we have almost 5 institutions. First is the Faculty of Dentals from Jamia Islamia. Then we have Maulana Azad Institute of Dental Sciences. Then we have All India Institute of Medical Science, Delhi. Then we have Rajendra Prasad Institute of Medical Sciences and the Banaras Hindu University. So we have almost all the sites has contributed various data points. With the various data points when I mention, it's like with some of the data come very specific to the the site. For example, BHU Banaras gave us a lot of data on the OSCC, which is the subtypes of the oral cancers. Then these are the primary contributors from the different sites. So when we start with the whole process, so it starts from the collecting the tissue samples. Then from the tissue sample, what we do is like we do a staining, the hematoxin and eosin staining, which typically does a staining of the nucleus in the tissue. So typically the pink color is actually the staining at the tissue level. And we have the nuclear staining done with the hemoglobin, which actually stains the nucleus. So if you see the normal tissue, you typically see that clearly that there is a difference structurally that you can see. So typically the main objective here is that, it's very easy to see from the what do you call as images, but this is just a snapshot of the tissue sections, which becomes very much, unique to each of the classes. We will show the hurdles that it comes when we are building the deep learning models. So first of the foremost thing that what we do is like stain normalization, because the staining was done in various different sites. So you will find that there will be stains level differences that will come. So first thing is that every before you build a deep learning algorithm, so first thing you have to do is the staining normalization of the tissues. So this shows the before staining and the after staining normalization, how the slides looks like. Once you have the stain normalization of the whole slide, then you start doing two activities, which is very, very important. The example that which I showed in the earlier, that once we have the whole slide image, you generate patches for your building the deep learning algorithms. So first you generate the patches, then also since deep learning algorithms typically needs more data points, also, generalization of the what do you call as, the images. For example, flipping is very, very important. For example, this is a nice example in deep learning where, if you have an image of a cat, you don't provide only one, what do you call as snapshot of the cat. You just rotate them in various scales so that, your algorithms can able to detect the cat from the image itself. So that process we do flipping. This is also put as a more of like a two-way we do a horizontal flipping and the vertical flipping, because if you do more, then the patch size increases. But we did not want it to have, because we have a lot of images to start with. So we had the patch size of around like 300 to 300 pixel. Then also we had the overlapping slide. So you generate from the whole slide image, you generate the small patches. And we had the complete workflow, which was there. Like, once we have the input of the patches, you have three different models. So first, the model it does is like, if you have a normal OSMF or the OSCC, then the model, let's say, it's a normal or the OSFF. If you have a OSCC, then we have a two different models where you first do a three-layer classification. So the first layer does only the well differentiating OSCC and the what do you call as medium moderated differentiating OSCC. The second model actually does again the poorly differentiating one. So we'll talk about this more in the coming slide. This is primarily we build the architecture so that the poorly differentiating and the moderately differentiating seems like very very close to each other, but but what we actually experience is that well differentiated and the moderately differentiated are looks like very similar to each other. So we built another model which can actually differentiate both the well differentiated and the moderated differentiated one. So we applied almost like many deep learning algorithms, and what we could actually find out is of those only the Inception V3 was able to perform very well in the validation data set. So if you see in the x-axis, this is the accuracy, and the blue actually is the for the during the training, and the orange depicts the validation. So this is one of the very prerequisites that we follow in our machine learning or in fact deep learning framework, that you start with a lot of patches, which is shown in the top. Then during the training, we use almost 144 patches. Then for testing, we use almost like 36,001, which is to do with only to build a first model, which is to build a normal OSMF, and then we have OSCC. Then the subgrouping of the OSCC was actually done again, which was composed of around like 286,000 data set. Then again, we splitted this 80-20 data points where you had 229,000 where for the training data set, and the testing was done for the validation was done for 57,000. And this is a typical framework that we follow for all our machine learning activities. So again, one of the aspects that we also want that other what do you call as the computer scientists or the community actually benefit from the activity that we have been doing. So we have made the data publicly available, where you guys can also start building models by your if you have infrastructure or the computational infrastructure with you. So you can take the download the data set and also build models on top of that, which we have not actually covered in our exercise. So you can follow this DOI, and I think I will share the slides also for your future activities whenever you want to have. So the most of the the work was funded by Ashoka University and also the Emphasis Lab, and also we have a generous funding from Rockefeller and also the Wellcome Trust DBT Alliance. Then we have the computing infrastructure and the support system was primarily from the Ashoka University and Emphasis. So for the hands-on session, I would request you all guys to come to this Google Meet link. I hope the organizers have shared you the Google link. So first we will start with the Amit's hands-on session, where Amit will be demonstrating how you can load high resolution whole slide image using your local system. Then we will have Akash. Akash will be showing you the how to generate from a big megapixel or gigapixel data to a more patch level image generation. And also he will be talking about how we do the stain normalization activity. So those are like part of the hands-on session that we have. Then at last we will be showing some work not related to oral cancer, but on the semantic segmentation. So if you have a image, and if you want to get, for example, from a tissue image, you want to find the stroma or the in fact cancer regions. So how we can use semantic segmentation with deep learning algorithms. So that will be covered by Somjit Bas. Thank you very much. So please come to the Google Meet link. I'll just. Are you able to join the Google link? Okay, I'll just admit.
Maybe you can.
Yeah, I'll just give. Let me just. Oh,
I'm not getting implemented on Wi-Fi anywhere.
okay. Can I make someone a host? Host. What's your name? You joined, correct? Rejakshi, no? I'll just make you the co-host. Is it done? Yeah, you have to just make others enter.
Yeah.
I'll just log out. So. I hope you guys can join the Google Meet link.
Join the Google Meet, you can leave the Zoom link and join the Google.
So
You
you are
can declare their file.
So I'll just get disconnect. Okay.
Thank you so much, sir, for such an amazing lecture. We learned a lot. And please everyone join the Google Meet for the hands-on session. The link is provided in the chat box.
Yeah. Okay, guys. We'll meet you in the G Meet link.
Ravi, can you hear me? Hi, Ravi, can you hear me? You are muted if you can hear me. I think you are the co-host. Am I audible? Not able to hear anything.
Video. Sir, hello. Hello, Ravi sir. Can you please disconnect your earphone?
Yeah, I have disconnected. Now you're
Yes,
audible.
yes. Okay, okay.
Give me a second. Yeah, let me know if my screen is visible.
Yes, sir. Clearly visible, sir, and you are audible and visible also.
Hello, am I audible?
Yeah,
Yes, ma'am, you are audible.
Okay. Hi, Ravi. This is Priyanka. I
Hi.
hope you can.
hello.
Hi, welcome to Amity. IT, am I audible?
Yeah, you are audible.
Okay, thank you. Please give us 2 minutes. We'll just introduce you and then we'll start.
Sure.
Okay. Hello, everyone. I would like to humbly invite Dr. Ravi Kumar Kushwaha, AI research scientist, Q.AI, for our next session. Mr. Ravi Kumar Kushwaha has been involved in training deep learning models for classification and segmentation of fractures on musculoskeletal X-rays, developing object detection models for 16 anatomy, writing production level code, and deploying the to Docker and much more. We are delighted to have you, sir, here. And now I request you to kindly start the session.
Yeah. Am I audible to everyone? Just a second. Yeah, am I audible to everyone?
Yes, sir, you are audible, sir.
Sure. Then I will start the presentation. Give me a second. Yeah, so like let's talk about AI and like let's keep this session interactive. Like you can pause me in between or like we can discuss about the things in general. So I have divided the presentation in 3 sessions, so 15 minutes each. And after each session, we will have a question and answer round. Yeah. So like, let's talk about why AI is everywhere now. Like what changed? 10 years before, like AI was not so dominant. Like we didn't use it in our daily lives. But why the machine has become so dominant and powerful? Like, can anyone, is anyone can tell ki what is your take on AI? We can have 2 minute short talk on this. Any candidate. Okay, if, none has an answer. Let's continue. So yeah, what's AI? So the first simple thing that comes to our mind is just a single image, like whether it is a cat or dog. So we need to distinguish, given the image of a cat and the image of a dog, which one is classified as cat and which one is classified as dog. And like since we have been using our smartphones for more than a decade now, so we have a lot of images on the internet. And we have in as seen in the figure, we have a lot of cat images and we have a lot of dog images. But how to conclude that this one is a cat? So what we do is that since we have taken a picture, each picture contains a pixel values. So that is an matrix of numerical values between 0 to 255. So you can use any function, as in like add all the pixels, subtract all the pixels, take the average of all the pixels, or do any sine or cos or non-linear function apply to all the functions, and at the end generate a value which will distinguish the cat images from the dog images. So that's the basic idea behind what we do in machine learning, particularly in computer vision. Just a second, I'm not able to move to the new slide. Uh, yeah. Yeah. So in earlier days of computer vision, what the task really involved is like, let's see, the left hand side is an Eiffel Tower, which is erect in shape, and the right one is kind of slightly distorted and scaled, but this is also an Eiffel Tower. So how we will tell that the right image is also Eiffel Tower corresponding to the left one? So what we do is that we map the vertices, like the top points, then we map the middle points, and other structure, similar structure, we map in both the images, and we come to the conclusion that yeah, it is a picture of an Eiffel Tower. Yeah. I guess there's some issue with the slideshow. Yeah. And like, let's say this is a cat image and we want to obtain all the edges of the cat. So the whiskers, the paws, and the tail, we have obtained the edges. But these were all based on the classical ML techniques, and which are not used in nowadays so much. Yeah. So what exactly happened? Like, why did this revolution, AI revolution, where did it came from? I guess you might be aware of the paper AlexNet. So what AlexNet did is it simplified image classification techniques. This was the birth of CNNs, popularly known as the convolutional neural networks. Yeah. So in convolutional neural networks, if we deep down, if we go into the maths of this, so it is the left-hand side is a pixel image, and we are having a kernel which is moving over the image and generating a value. So with the help of this technique, CNNs got popular, and this was the first time like humans, like the machines became powerful than the humans. And also thanks to the powerful GPUs required for training these models, we are now able to achieve good accuracy on the ImageNet classification. Yeah. Yeah. And now the image recognition tasks, we have divided it into three main tasks. The first was the classification. So classification is just like predicting one value at the end. Given a input cat image, we pass it through the convolutional neural networks models, max pool it, and at the end obtain a value, which will tell either it is a cat or not. But let's say you have a tumor. This scan contains a tumor that the doctor has diagnosed, but where exactly is that tumor? How to localize it? So we need something called as segmentation. To accurately in the left-hand image, you can see the tumor has been highlighted. So not only telling where the tumor is, not only telling if there is a tumor, but also telling you the location of the tumor. Yeah. So this was the general unit model. So what you can see on the left-hand side is that a man is dancing, and on the right-hand side, we have been generating a masks of all the pixels involving that person. Yeah. So unlike classification, we don't need a single yes or no value at the end, but each pixel is marked as if it contains the tumor or not. So here you can see at the end, we have the output size equivalent to the input size, because each pixel you need to tell whether there is a tumor or not. Like this is a 3D MRI scan. And after applying the unit model, we will get the tumor segmentation. So for each pixel, you will tell either there is a tumor or there is not a tumor. This will be needed. The third kind of image classification, third kind of image recognition technique is the object detection. So here in this video, you can see like there are several objects like the cap, the mug, and they are being identified by our neural network model. So why object detection is needed when you have a segmentation model? If you can classify if you can localize the tumor so well, why do you need an object detection model? So object detection model is a lighter model because it has lesser parameters to train, and at the end, they just give the rectangular boundary as output. It doesn't draw a heat map for every pixel. So it is faster to train with less memory requirement and very easy to annotate. Yeah. So here, if we train this model to tell about the tumor, it is not giving the precise locations, but it is giving a good rough boundary around the brain cells which contain a tumor. So it is easy to annotate and train also with faster implementation. This is a blood count, blood cells like RBCs and WBCs. So instead of segmenting it pixel by pixel, we have just drawn a rectangular boundary around it. So this makes the work easier than the segmentation model, but the catch is that the accuracy is not so obvious as the segmentation models. In this line of research, there was a paper called YOLO. Its full form is You Only Look Once. So what you do is that you divide an image into an S by S grid, and in every grid, you search for boxes which can contain an object. So here in this image, you can see if the dog is there, there is a cycle and there is a vehicle at the end. So this is easier implementation than the 3D unit model. So this was the architecture. Yeah. And YOLO versions have been keep on updating over the years. So this is the newest version, YOLO version 8. So you can see how accurate the model is, and it can perform in real time as well. Yeah. So we discussed about the 3 models. So if anyone has a question or something to say, we can wait for a few minutes and take that on. Okay, we will now proceed to the second section. So yeah, what problems are we facing in medical domain? If AI is taking over so rapidly, then it should take in medical domain as well. But there are a few challenges involved in medical images. First one is the limited data availability. We don't have data as in the internet. You can see there are variety of images, but for medical images with some disease, you will have very limited availability. And also the annotation is very expensive. If you want to get the tumor marked by a radiologist or by a doctor, so they will charge a heavy amount for each annotation, for each scan they tell. Yeah. Also, there is an issue of data privacy. The hospitals don't want the data to be used for some AI model or some research. So this is also an issue why we have limited data. And the very common issue that we are seeing is the data bias and diversity. Let's say you have taken a fracture data set which contains fracture from the Indian population, but what about the US and the African population? So it may happen the model that generalized to the Indian population may not work well with the other geographies. So we have these problems that we need to sort out in order to obtain good accuracy on the medical image set. Yeah. So the first problem is the class imbalance problem. What this tells is that 10% of the classes account for 93% of the data. So what about the remaining 90%? They have very less representation. For example, pneumonia on chest x-rays are easily available in abundance. You can easily get the data, train your model. Whereas a rare genetic disorder, you won't have much data since they are rare. Yeah. Also talking about the annotations. So here in this image, you can see this is a 3D MRI images. So a 3D MRI image is projected into 2D slices. So there are so many slices, and we needed to obtain the blood clot that the brain has. So a annotator, a doctor here has marked the blood clot on all these slices, but you can yourself realize that how tedious this work is, and this is just for one MRI. This will take an hour for a doctor to analyze all the blood clots and mark it here. So this will be very time consuming. It will also be error prone because the doctor won't be in a good state of mind every time to annotate these images. And it will be very expensive since it is time consuming and you require someone very skilled to label these data. So what solutions do we have? One easy solution is data augmentation. So let's say in the left upper image is of a lung. You can scale it, clip it, resize it, and you can distort it and make another image of the lung. So basically we have one image, and from one image we have obtained 8 different images. So we will use all of these for training. Now let's say the left one has a lung cancer. So all of these images will have a cancerous cell, and we can train our model accordingly. So this is to generate more data. Yeah. So data augmentation basically involves, as you can see in the picture, there's a butterfly here. So butterfly, we can have the original image, we can detexturize it, we can remove the color, we can enhance the edge. Like in the fourth image, you can see the edges are broader. Then you can have the edge map only, not taking the interior parts, only the edge. And the very common is the flip and rotate part. You can rotate it at an angle, you can rotate it, you can flip it vertically or horizontally, and you can obtain new data. Yeah. One more thing, one new concept in medical imaging is the self-supervised learning. So first let's see what is supervised learning. So in supervised learning, let's say you have an image of a tree and you pass through some convolutional neural network, and it predicts dog with some probability, tree, and table with the highest probability. But since you know this is a tree, you will have a loss function which will tell that no, this prediction is wrong. So you need to change the prediction so that the prediction is close to tree, closest to the tree, and not close to dog and table. So this is a supervised learning, that the input has a given output and we try to match that output, that label. But in many cases, yeah. So in medical imaging, it is very hard to obtain data with accurate labeling. So what we do is self-supervised learning. Here we have labels, but we want to train our models without labels also. And yeah, this is a very hot topic of research. Like many companies like Facebook, Google, they are working in this domain specifically. So what is self-supervision? So in self-supervision, we obtain labels from the data itself by using a semi-automatic process, and we predict part of the data from other parts. Let me show you through an example. Yeah. So this is a $1 note. On the right hand, it is the actual one, and in the left hand, it is just written ONE1. There is a numerical representation 1, and there is an image of George Washington in the image. So if I give you to draw or recall, you give what is $1, what do you recall? So only taking those parameters or those features that are necessary and disregarding all these leaves are there, then there's some random number or signature. We all disregard these things and just take the images, take the features which are meaningful. Yeah. But how do you train without labels? Earlier in the tree example, we can see that there was a loss associated if the output of the neural network didn't predict tree as output, then we had a loss function. But what to do here in case where there is no label? So what we do is that we take an image of a dog, and the other image is an augmented version. Here it is decolorized and flipped version of the same dog. So what we want, that when it passes through a neural network, we want to have the feature embeddings of these two images as close as possible. Yeah. So we wanted the augmented version of the same image constitute a positive example. So here you can see this is a feature embedding space where the blue are the positive samples. They are closely placed, whereas the green and violet ones are relatively far. So we want that all the dog images be closely placed, and let's say there is a cat image that should be very far from the dog. Yeah. So cropped sections from different image constitute a negative sample. So cropped image from a dog and cat, they should behave differently and they should be well separated in the feature space. Yeah. So the advantages of self-supervised learning is that it can leverage a large amount of unlabeled data. So let's say you have a label of a dog of only 10 images, but I give you 100 images of dog. So from similarity matrix, you can see that all the images are for dog only. Yeah. It also enables models to learn relevant features from the data distribution that might differ from the target domain. So let's say you have a deployment in some other country, but the data was trained in Indian context. still SSL will able to make generate more data, augment it so that it can generalize to wider geographies. And of course, the problem was to reduce annotation cost and errors. So that is solved. So SSL helps us to to learn the representations from a limited data set. But one more thing we can do is to generate more data. Last time it was just the flipped, augmented, rescaled, reshaped version or cropped version. But what about generating more data? So here in the image, the left hand side, you can see on the top, there is a real chest X-ray. And on the bottom, that is a chest X-ray with visible pleural effusion. So we wanted to train our model by generating more data. What we did in the upper columns, in the upper row, we had made just normal X-rays, more normal X-rays, whereas in the bottom one, we had made the lung X-ray with pleural effusion. Like you can see at the bottom, there is some liquid contained in the lungs. So we are generating more data through GANs, that is generative adversarial network, and diffusion models. These are also area of hot research right now. Yeah. So we generate more data. Like here we can generate the tissue data. This is the brain cells and the 3D brain data we have generated. So we can generate data in any domain, medical or other fields as well. Yeah. Now the next topic is the zero shot learning. What if we don't have any data for a disease? Like let's say you have a very rare disease and it is hard to find even a single occurrence of that disease. So what we do? So here we explore multimodality. So multimodality is like one modality is the images and the another modality can be the text. So generally we have an X-ray associated with a report. So we combine the different information from different types of data and use the reports associated with the scans. Yeah. So for example, we can see here we have the medical image data set and we have the medical test data set. So there are different types of lung X-rays and there are different type of reports as well. So here the first image, in this image, the report says that the new left lower lobe opacity, there is an opacity in the image suggestive of left lower pneumonia. There is an image on the right which tells that there is an opacity, but this opacity is concerning for either pleural mass or extra pleural mass. Left one contains pneumonia, right one contains an opacity with pleural mass. So if let's say we don't have any data for opacity, if we have an or any other abnormality, we don't have any data. So what we do is we try to map like these both are different images and these both are different problem, but we try to combine a problem which is seen in one of the images. So that's how we find data for some rare disease. We in the feature embedding, they should have some kind of similar representation. So this is how zero shot learning is done. Yeah. So we have come to the end of the second session. If anyone has any question or would like to give feedback about any of the models, we can talk. Yeah, moving on to the third part of the session. So what till now we have a separate image for we have a separate model for images, separate model for videos. Like here you can see the first image is the cat image, second is a cat video, and third is a 3D representation of cat. So the machine learning models were different in all these modalities. So what we want to do is to why to have separate models for image, video, and 3D. So there is an omnivore model that has been developed by Facebook AI Research, popularly called as FAIR. So they combine all these modalities and predict and give us a general output. And they have termed this as omnivore model. So here, like let's say you have an input, you have a video, and you have a single view 3D. So from the video, you can have multiple slices. So they will correspond to multiple images. in the single view 3D, you can have a depth map, you can have an RGB map for the projections. So what we do is that we take one model and we feed all the images, video, as well as the 3D input to the same model, and at the end we generate a single output. So this is how we train a model, multimodality omnivore model. Yeah, we are focused on vision, like computer vision regarding images. But what about NLP? And why we are discussing NLP? So NLP is the natural language processing, which is applied to sequence models. As you can see here, the on the left hand side, we have a French phrase, which the model tries to convert in English. So this model tells that this probably is I am a student. So this is a sequence to sequence model. So from French text, we have generated English text. And why we are discussing about NLP, I will discuss in a bit. Yeah. So NLP also helps in like sequence completion. Like you have to fill in the blanks. I like cats more than. So here the last word can be anything, but the model has learned that it has to be a noun. It has to be similar to cats. Here they are not predicting it as plants, or here they are not predicting as a word, let's say cry. So the model has learned how to get contextual information from the input and predict a final output. So here it's I like cats more than dogs. So this word has come up after training of the model. Yeah. So there's a research going on in transformer domain. So transformer first came in the NLP, and this has popularized the attention mechanism. There was a very famous paper called Attention is All You Need. So what it does is that it focuses on different part of the input sentences, making them adapt to capturing contextual relationships and long range dependency data. Like let's say in this example, as I have earlier told, the last word, the model has figured out that it needs to be an animal name, and it needs to be a noun at first, and then it needs to be an animal name. So that the model has learned. So thanks to transformers, this has been possible. And what are the other uses of transformers? Yeah. So first, transformers. So Chat GPT, I am aware that everyone would have been using it by now. So Chat GPT. So GPT is generative pre-trained transformer. Here you can ask any question, and like it is just kind of a fill in the blanks. Earlier in this question, it was a small fill in the blank, like I like cats more than dogs, one word. Here it is that you have asked a small question and they have generated an output. So this is a transformer only. And working on the transformer, we can see, yeah, like here, the first student, this is French, J, has gone to the encoder of the transformer, followed by decoder. And here the output has been come as I. The second, Sui of French has gone into the encoder and then fed into the decoders, which is predicting that I am a, and last student, and also the end of sentence. So this is how a transformer works. But yeah, it is a sequence to sequence model. Like why we are discussing this, because due to transformers immense popularity and the self-attention mechanism that we have seen, there was a paper on vision transformers. So vision, but how to convert an image into 1D. So what we did is that here's an image of a tree. So divide it into 9 patches, flatten them, and project them as a sequence. Just like any text sequence, we are projecting this images after dividing them into 9 patches and flattening them. We are feeding this to a transformer encoder, just in the case of NLP. And at the end, we are giving one output as tree, and the model is learning from that. So the vision transformers, analogous to the transformers in NLP, they have also gained immense popularity and have proved useful on a variety of projects. Yeah. And transformers, not only they give us a classification output, as in the case here, they are giving a MLP head, that is multi-layer perceptron at the end, and giving predicting a class as output. They also give us segmentation maps. So let's say here you can see a dog is standing in the image. This transformer has learned where the dog is exactly. And hence we can see a attention map. And from this attention map, they have cropped out that in the image, the object, that is dog, is present here. Same in the bottom image, you can see there's a bird flying over the ocean, but we need to focus on the object bird. So the attention mask has been generated across bird, and at the end we have got a segmentation output. So this is all with the help of transformers. Yeah. Coming to the last part of the presentation. So hands-on, like clearly we have seen there are so many topics and so much research that has already been done in the deep learning domain, particularly in the medical imaging. So as a student, like if you are a fresher student, so how to start working on these projects? This is a lot of information to digest and also will require a lot of effort from your side. But what are the basic steps? How will you start working on these topics? Yeah. So first thing is Python. It is absolutely necessary in today's field, be it academic research or industrial research. You have to be very good in Python. Yeah, there's a message in the chat. Yeah. So multimodality is that, just a second. Yeah. So multimodality refers to, just a second. Yeah. So in this image, you can see we have a lot of images and we have a lot of text reports. So one modality is the images, another modality is the text report. So we want to combine both of these information and train a model. So this is called multimodality. And also the omnivore model here also, one is image, one is video, and one is 3D representation. So these all come under multimodality, since all of them are different type of inputs. I hope this clear answers your question. Yeah, we were discussing as the student, how should you start? So you need to be good and proficient at Python, and you have to learn about the basic libraries like the NumPy, Pandas, PyTorch, which is useful in implementing all the deep learning techniques. So Python is a must and you should start practicing. Yeah, another good source of learning is Kaggle. I guess you must be aware of Kaggle website, and the Kaggle competitions are there. So you can start with, yeah. Yeah, so Kaggle, you can get started with like, let's say, small classification or segmentation problems. So you will have access to the data there. You will have also access to some basic codes, as in how to write the code. As also you will be evaluated on your accuracy. If your accuracy is low, you need to like change your model, or you need to perform some augmentation, tweak in, and get a good performance on the data set. So Kaggle is a very good resource to start working on deep learning projects. So this has plenty of projects on every field, like be it self-supervised learning, be it reinforcement learning, or be it a simple classification where you need to just get the images classified as whether which number is written. Like this is a digit recognizer, also called MNIST dataset. Yeah. Also, these were the first basic tools, as in Python and Kaggle. But what about the latest research that is going on in the deep learning field? So the first thing that, so the third thing, like after Python, after you're familiar with Kaggle, then start reading the famous research papers. like unit, as we discussed for the segmentations, is a convolutional neural network for biomedical image segmentation. Yeah, we will take the another question. Okay. Yeah, then there is contrastive learning from unpaired medical images and text. So this is the second one. This is a multimodality training of the models. And attention is all you need is a transformer model. You can search for the other research papers which are now state of the art. You can read them, and these involves a lot of maths. So also be prepared to dive deep in the mathematical domain and understand what is exactly being done. So this will contain a lot of maths, the research papers. Yeah. And also, once you have read the research papers, you can find the repositories online for all these above and the other famous research papers. So there's repositories are easily available on GitHub. So you can just clone the repository and you can start implementing it on your own projects. So you need first to read the papers and then find the repositories and start training. Yeah, also one very famous question for students is that where they will be able to train the model. Like GPUs are costly. If your laptop doesn't has a GPU, what to do? So I guess most of you would have heard about Google Colab. So Google Colab will give you a free GPUs for limited hours where you can train your model. Also, there is Colab Pro where you can pay and have the GPU for a longer time. Yeah. So we reached the end of the presentation, and like we discussed all the modalities required in medical imaging and how to start. If you have any question, then please let me know.
Hello, sir. I have a very basic question. That when we are working with the images, and can we work? What's the limit population of images we can train a model on?
Your voice is not clear, like it is echoing. Can you please repeat?
Yes, sir. So my question is that what's the limited number of images that we need to train a model on? What should be the minimum population of the images that we have to work on image data?
If you have a like a classification problem, if you have a segmentation problem, then you can work with less data also. Like, but let's say having 1,000 to 5,000 will be a good initial approach. But for like in industry level, we work on millions of data, like 100K, 1,000K is normal for us. But for finding something very common like pneumonia, you can have even 1,000 to 2,000 images will be fine for that.
Thank you so much, sir. Does anyone else have any questions? I think no one else has any question. So we'll end the session here.
Yeah, I will stop sharing.
Thank you so much, sir, for such an exceptional lecture. We were really intrigued into that image segmentation and training models. And so if anyone else has any question, please ask right now. I don't think anyone else has any question. So for now, we'll have a break and we'll meet again here at 6 p.m. sharp.
Thank you so much, Ravi, for accepting the invite and being here with the students. I'm sure they must be having a nice time listening to the lecture, and it was so elaborate to understand the basics of imaging and segmentation and everything around it. So thank you once again. And we would definitely love to have you on campus sometime soon. So next time, maybe whenever we are able to call you, we will definitely get back.
Thank you. It was a pleasure to present at Amity.
Yeah.
Yeah. Okay, if you have nothing to discuss, then we can discuss. Thank you for giving me the opportunity to present.
Thank you so much.
You are welcome.
All right, all the students, participants, you are kindly requested to join back again at sharp 5:50 because we are going to start the session at 6 p.m. with IUP-UI speakers. We have 3 speakers lined up for you, and they will be also touching topics around medical imaging. So I would request all the participants to please join at 5:50 so that we can start the next session on time. Thank you for now.